I would argue and I would answer that anyone who's studying RNA in any way, shape, or form should now put the lens and filter, or whatever you want to call it, of epitranscriptome into their thinking. There's been some recent discoveries that are showing how valuable RNA modifications in the epitranscriptome and the RNome are for diagnostics, complex information-rich systems that tell you a lot about disease risk, many different kinds of diseases.
Well, it's great to see you again, Pete. It's nice to have a little opportunity to ask you some questions that, we don't necessarily get to in our other meetings. So I want to ask you first, you went from an early research focus in DNA damage and DNA epigenetics and shifted more towards RNA modifications. What led you to make that change?
Well, I'm a nucleic acid chemist from the get-go, and I had an opportunity to jump into this field not knowing much about the RNA modifications, but it just seemed like such an exciting new area with so many possibilities. And as an MD, I also thought, what a potential cause for so many diseases.
Mm-hmm. So what were some of the pivotal discoveries from your lab, in the early days that helped you to solidify your focus on epitranscriptomics?
Really, it was collaborating with Tom Bagley at the University of Albany and doing a multi-omics analysis of tRNAs and tRNA modifications in yeast and looking at how they change as a function of stress. And putting all that together led to a discovery of a pathway involving reprogramming of the tRNAs that leads to selective translation of stress response genes. That's the whole story behind the work we're doing.
What we wanted to do was to put these tools in the hands of everyone. And so, as you well know, the Alida Bio Epiplex tools are enrichment-based barcoding methods that are designed to be really sensitive and to, allow you to work with things like precious clinical samples and to measure changes in the RNA modification abundance at each individual locus. So how do you see the field growing as we, continue to have great academic discoveries and more emergence of commercial tools that can allow anyone who's doing RNA sequencing to work RNA modifications into their basic or applied research?
Yeah, it's a great question. And I think the biggest challenges are simply to map 55 modifications in 250,000 different types of RNA that are in a cell. And you can do that with mass spec, next-gen sequencing, nanopore sequencing, tools like Alida Bio's Epiplex kit now to be able to tell you which RNAs have those modifications and ballpark where they're located in the RNA. Super critical tools to put in the hands of researchers at every level, whether they're experienced or inexperienced, and start mining the epitranscriptome in health and disease.
So, with some of these commercial tools, of course, the Alida Bio is one. Some companies are trying to make epitranscriptomics more accessible to RNA researchers in a general sense. How do you see that having an impact on the field as anyone who is doing RNA sequencing starts to be able to sequence the modifications too?
It's going to have a huge impact because people are going to discover things that we never knew previously, simply by asking a new question, measuring it with some precision and some accuracy, and coming to new discoveries about tissues and diseases we had no knowledge about with the epitranscriptome.
Mm-hmm. What are some of the biggest unanswered questions in epitranscriptomics today?
The biggest unanswered questions are where are they and in which RNAs and how are they changing in states of disease and pathology and even just in normal growth and development.
Do you think that there's a lot of untapped opportunity out there to directly manipulate RNA modifying pathways therapeutically for human health benefit?
Yes. Short answer to that is yes with an exclamation point. STORM it typifies this. And this is the interesting part of the human RNome project. With the Human Genome Project, there were no companies out there exploiting this. There's already dozens of companies doing RNA modifications, therapeutics, all of that before we've even established the field. So the impact of RNA modifications for therapeutic targets is already huge. You think of the fact that there's 100 or so different enzymes that put in 55 modifications. We know that there are 100 or more diseases caused by alterations in those enzymes and in the modifications. So there you have dozens and dozens of potential targets.
And how do you envision the future of RNA epigenetics in basic science, diagnostics, and therapeutics?
Well, I think there's been some recent discoveries that are showing how valuable RNA modifications in the epitranscriptome and the RNome are for diagnostics, complex information-rich systems that tell you a lot about disease risk, many different kinds of diseases. So I think that's super promising. And then you've got the cause and correlations of RNA modifications with hundreds of human diseases. You now have therapeutic targets to go after. So huge opportunities in both diagnostics and therapeutics. And ultimately, it would be great to get, for example, hospital labs using technologies that Alida and other folks develop to quantify epitranscriptomes and RNAs as a diagnostic biomarker for cancer diagnosis or to follow treatment. Any of a number of different risk biomarkers that you could measure in blood or urine or feces or any accessible compartment. That'd be a major biomedical breakthrough to have that kind of technology out there. I would argue, and I would answer, that anyone who's studying RNA in any way, shape, or form should now put the lens and filter, or whatever you want to call it, of epitranscriptome into their thinking. Hit the literature, start reading, learning, going to conferences. And this is a problem of gray-haired people who are set in their ways and not about to learn a whole lot of new stuff. This is a younger person's kind of adventure. Get out there. If you're doing any kind of RNA work, you'd be silly not to go see if there are modifications that could be involved in the questions you're asking.
Well, it sounds like there's a lot of exciting opportunity ahead. Thanks so much for the chance to ask you a couple questions, Pete.
You're welcome.